Hello, my name is Matthew Tatz, and today I'm going to be doing a lecture over avian salmonellosis. A brief overview of what we'll be covering today. We're going to look at an introduction to Salmonella enterica, history over this pathogen, avian salmonellosis epidemiology, passerines, which is feeder birds, and management. Okay, so what is Salmonella? Salmonella is a bacteria from the genus Salmonella. The species of Salmonella that we're concerned with today is Salmonella enterica. It is a zoonotic bacteria, meaning it's spread through animals to people or animals to other animals. This pathogen infects a diversity of hosts, including birds, pigs, people, etc. And it has 2,300 serotypes. Now, serotypes are subsets of this bacteria that are slightly different from one another. So Salmonella enterica, the serovar that we're focused on today will be Typhimurium. Typhimurium has over 300 different phage types, which is just another more specific level of classification for this bacteria. And some of the phage types include DT2, DT99 in pigeons, DT13 and 14 in poultry and ducks, and DT40 in finch sparrow, which is also the greatest phage type that we see in most wild birds, which is what we are concerned with today. So a history over Salmonella typhimurium, which we're going to call ST from here on out. It's considered an emerging disease because the presence of this disease in wild birds has exponentially increased over the past 40 years. This is due to artificial feeding, so there's been a surge in the amount of bird feeders being used in this time. So in 1957, the first time Typhimurium was reported in wild birds. Hudson and Tudor found salmonellosis in house sparrows, starlings, brown-headed cowbirds, and rusty blackbirds in northeastern US. These researchers suggested these outbreaks were related to birds concentrating at feeders during the winter, which we will talk more about in detail later on in this presentation. So we're going to be looking at the epidemiology of avian salmonellosis, which includes who is affected, where are species being affected, when are they being infected, what is the infection and why. But we're going to look at the what for barn owls, red-tailed hawks, crows, turkey vultures. And the reason they are infected is because they are eating the infected prey directly. ST is found in the intestinal tract, so when they are consuming other birds, they are directly getting ST themselves. Gull populations have increased over the past 30 years due to why? Scavenging activities, so scavenging around garbage dumps, sewage outlets, landfills. If you go to the beach, they're eating all of our trash, and that is where they are getting ST. Waterfowl are generally not infected by Salmonella, although we have found recent years that if they are living in contaminated water, so if waste is contaminating water, then they can develop ST, especially in juvenile birds. And then pigeons, such as rock doves. One study showed that 30% of pigeon feces, they detected Salmonella typhimurium in 15 of 17 cases. And this is maintained in their population by feces. So there's some rock doves. Okay, so the main species we're concerned with today are passerines. Passerines are any bird of the order Passeriformes. This accounts for greater than 50% of all bird species, and they're generally referred to as songbirds. So, which age of songbirds are mostly impacted by ST? As with most species, younger birds are more susceptible because their intestinal tracts have not developed, so they're more prone to developing ST, salmonellosis. In sick and dead songbirds, sick, moribund, or dead songbirds tend to attract, which causes them to further shed ST. Two researchers, Wobesser and Finlayson, isolated ST in 90% of sick and dead sparrows. Who's not as affected? If you're a healthy songbird, Tizard, fish, and ptarmigan only isolated ST in 15% of that same population of house sparrows. So it's generally low prevalence if you're a healthy species, healthy animal. Okay, so where is this happening? Generally, birds that congregate are more prone to spread ST due to feces exposure. So examples of congregation, the hooded crane is a species of crane found in Izumi, Japan. Every winter, they all congregate in Izumi, and that accounts for 70% of their population that gathers, which can lead to transfer of the disease more easily, transfer of the bacteria more easily. Another example are black skimmers. They're ground nesting seabirds that form dense colonies, and so ST would be able to travel very quickly through them due to the close proximity. Where else? The main place that ST is infecting other birds from infected birds is through bird feeders. So feeding songbirds is one of the greatest contributors to spreading avian salmonellosis. Bird feeders are a significant source of food, such as peanuts and sunflower seeds, which are very high energy. They're very significant in the winter when birds have a harder time foraging. So they actually do rely on these bird feeders. Large quantities of feces are deposited in and around these feeders, which causes the bacteria to spread. One evolutionary advantage of birds is that they poop and then they fly away and they don't return to where the poop is, and so they're not being exposed to bacteria such as ST. But when you have bird feeders and the birds are all congregating, they are returning to where they are pooping and where other birds are pooping, and so that's what's going to cause the bacteria to spread more quickly. So generally, if you have more food at a feeder, you're going to have greater death because you're having more birds congregate there over longer periods of time. 3 criteria for this. 1, appropriate species, adapted phage type of ST. So you need to be a bird that is prone to getting ST, like the house sparrow. Dense populations of birds need to happen. So in bird feeders, that is an example, or the cranes in the last example. And stress factors. So as you can see in the picture, when you have only one feeder and the birds are competing for the same resource, that causes higher stress. Areas on the graph are ST isolated in birds, and this is one single strand, and you can see that it's very widespread across the country. So it's not just confined to small areas. Okay, so when are these outbreaks happening? Most outbreaks occur in the winter. Bird feeders in the winter have become a significant source of food, like I mentioned. Some examples include the winter of 1992 to 93, pine siskins, evening grosbeak, sparrows, finches. There was unusually high rain and snow that year, which caused many birds to consult the feeders, which led to a big outbreak of ST. Winter of 97 to 98, 73 passerine cases were reported. In the prior 10 years, only 2 to 3 cases were reported, so there was a big outbreak there. In the winter of 99 to 2000, it was a cold, rainy summer. In Sweden, only 20% of 32,000 acres of linseed was harvested. So a large number of these birds survived in the autumn and the early winter. But when it came later in the winter, the birds all congregated at feeders, which caused ST to transmit, and that caused great death among those large populations. And then in Hokkaido, Japan, in 2005 and 2006, and then later in 08 and 09, there was a mass mortality event of Eurasian tree sparrows. There was 202 deaths and 100 incidents at 94 sites. 11 of these 13 sites was related to wild bird feeding, and 88% was found to be DT40 in the fecal samples. Okay, so these 2 graphs just show that it's generally happening later in winter. So the graph on the left, you can see that death-related incidents for ST and carcasses are occurring highly in January and very highly in February. And similarly, in the graph on the right, you can see that in February and then March, you have a high incidence of ST exposure and bird death. So why is this happening? ST is transferred through feces or ingestion. So bird feeders, they're exposed to poop and they are contracting the bacteria that way, or the birds can live or feed in contaminated water, such as some certain shorebirds, or if you have unclean bird baths, they can also contract it that way. Okay, so a very brief, quick review of the pathology of ST. Salmonella is found within the intestine. First, salmonella cells attach to the intestinal epithelium, which is just a type of animal tissue. Then the bacteria invades that tissue, as seen here. The bacteria can also be taken directly by dendritic cells into the submucosa. Salmonella replicates inside of the cytoplasm if not directly taken, as seen here. Bacteria is released to the submucosa. Bacteria is internalized within phagocytes, and then with phagocytes, they disseminate through the lymphs to the bloodstream, and then it goes all over the organism's body. So finally, management. How can we reduce contamination? Well, bird feeder maintenance is huge. You should limit the amount of feed used to prevent crowding of birds. You should keep your feeders clean so that fecal contamination is low. Use bird feeders with smaller platforms. So as seen here, this limits the amount of birds that can gather on a feeder, which would reduce stress, and you're not having so much fecal contamination build up at the bottom of the feeder. Here it's just on the floor. Now that's tricky because if poop gathers on the floor and seeds are falling onto the floor, that can cause greater exposure. Also, providing several bird feeders reduces stress by reducing the amount of competition at one source of food. Also, cleaning your bird baths so that water is not contaminated. Furthermore, waste management for things like shorebirds. Waste management companies should keep waste contained. You should prevent waste from leaving containment and prevent animals from entering facilities. So as seen here, there's a white ibis in a dumpster where it could easily